Hello everyone. Welcome to Reproducibility for Everyone for I Love Methods Week. Next, please. My name is Susann. I'm a plant biologist and lecturer at the TU Dresden in Germany. I have a couple of students, hopefully, of mine joining today. Yeah. Next.
Sorry.
We, Reproducibility for Everyone, we are a community of scientists in academia and industry who want to improve reproducibility of science. So we have members all over the world. We are a great community, big community. Everyone's welcome. So if you want to join us, maybe host one of these workshops in your own group, in your own kind of research bubble, just reach out to us, get back to us. Next, please. So we had a lot of great funders in the past. Currently, we have an NIH grant that's keeping us running. So thanks a lot for those already. Next, please. And if you want to get a bit more context about our initiative, we also wrote a paper on what we are doing. There are some resources in it. It's a preprint and it's a peer-reviewed paper. Next, please. So if you visit the shared notes, you can find all of the links to that there as well. We have a code of conduct. I'm not going to read this out. We just want you to please be at the best behavior when you are in this interactive workshop. And so if there's anything that is of your concern, just reach out to us. We have this pad where you can see that you see the short notes link there at the bottom of all of the slides. And in this pad, we're going to do the communication. So instead of doing the Zoom chat, instead of using the Zoom chat function, just use the pad. It's structured with all of the sections. There it is. We have already some good engagement going on. So people are telling us how they are, how much coffee they need for the day. But you see all of the sections there, and if there's questions popping up, we're going to address them at the end of the whole workshop, and then we can see accordingly where you had your questions. So please use that. And if you have liked something, then plus one it for your favorites. Okay, next, please. So what does reproducibility matter to you? Next, please. Or why does it matter to you? We ask you, and I just freshly copy pasted this. So we had 109 people registering for this workshop. So you see here, we ask if you had ever issues reproducing own or someone else's research. And we have a lot of early career researchers, undergraduate students also here who probably haven't done research yet. So they maybe answered roughly, they probably answered, I never had issues doing that. But you see that the majority of people who answered here said, yeah, I had issues reproducing someone else's or my own research. That's a very common topic that we see. I guess this is why you're here, right? In this workshop. Next, please. And so what we're going to do today is we're going to go over a sort of reproducibility framework. So we're going to go over good scientific practice with the lens of how to make it reproducible and give you some tools. And we hope that this is a good starting point for your lifelong or career-long journey. So there's going to be a lot of information probably, but you don't have to kind of use all of it right now. Next, please. So we will look at a little bit now what reproducibility means, and then we have this kind of grouped a little bit in the tools grouped in different kind of categories: organization, documentation, analysis, dissemination. So this, you're going to see that structure throughout. Next, please. Next. So definition-wise, when we talk about reproducible research, what we mean by that is that the authors of a manuscript provide all of the necessary data and the computer codes to run the analysis again, and thereby we are able to recreate the results as readers also. A replication is a study, a separate study that arrives at the same scientific findings as another study, but they collected new data and completed new analysis. Next, please. So there are some terminology, different kind of modes of reproducibility. And so the absolute cornerstone of each scientific project is reproducibility, where you do your own research again. So you use maybe the same data again, address the same methods, you repeat your experiments, and they still always come to the same conclusion. So that's reproducible research. That's the absolute cornerstone, which we need, right? So you see here, there's also some other flavors to that. If someone would use your data, but they employ a different method, come to the same conclusion, it would be robustness. And then, of course, in the end, we want to have a generalizability. So we come to the same findings, even if we collect new data, tweak the methods a little bit. So we talk about reproducibility here, so about the absolute kind of minimum things you should do to make your work reproducible. Next, please. So is it all that matters, reproducibility for research? Next, please. Well, of course it's not. We always kind of assume people work rigorously. So scientific rigor is kind of the absolute basis of what you do, good scientific practice. People have an understanding of error analysis, how to design experiments right. They are logically structured, right? So you have a good understanding of probability statistics and you are intellectually honest. Next, please. So this is kind of what we assume or what we think that you are doing, or yeah, you have this kind of knowledge. So probably there's a lot of things you're going to learn here. So every little bit helps, and it's really, you don't have to be perfect. So everyone starts in this journey somewhere, and what we aim for is to teach you how to do kind of transparent and open science. And that's kind of what your guiding principles should be in your scientific career. Next, please. So there's a lot of different factors who are decreasing reproducibility, and we just want to go over some of them. Next, please. So we grouped them a little bit into the technical factors. So if you work with cell lines or so, which could be contaminated, antibodies are not validated, these kind of things can decrease reproducibility. Misunderstanding of statistics, so study design and statistics errors, or human factors like insufficient detail about methods and materials, these all decrease reproducibility. And these are usually without people maybe realizing that. What you see here, though, in the, let me see if I get the pointer across. No, I cannot. What you see here, there's also in the middle, there's this lack of training. There's hawking. Hawking is scientific misconduct. So this is hypothesizing after the results are known. This is a practice that is not intellectually honest. So that's something people do, well, and they sometimes get away with it. So that's not good scientific practice. And also p-hacking. So doing an experiment with large numbers again and again until the outcomes fall under this magical number of like p equals 0.05. And so it's statistically significant probably. But again, this is not giving us the true results. So there are some things that people do out of bad practice. But most of these things you see here are like also the external factors. These are things we cannot really influence probably, right? So the rewards for surprising results, publication bias and high impact journals, these kind of things are really hard to influence on as a personal or on a personal kind of level. So we don't focus on that. We focus more on the human factors we can control. Next, please. And so reproducibility came out of this kind of open science movement wave in a way. So there are some disciplines like psychology and others where there were some huge cases of scientific fraud and kind of things where science could not be replicated, huge findings could not be replicated with today's methods. And so we want to go back to say, okay, reproducible research really hinges on the understanding of how experiments were performed and how data were acquired. So we need to have this information as a person reading the scientific papers of someone else to be able to also trust the results because we know exactly what people have done. And so there are two very critical elements to improve reproducibility. The first is an increased transparency and a sharing of research reagents themselves, so the physical things, the methods and the data. And the second is the accelerated sharing of research in which come in the form of preprints. So this will accelerate science and makes it more transparent and open. Next, please. So where's your greatest potential for growth? That's a question when I do this in person. I usually let people write on that. But here we ask you, what are major improvements you can think that could lead to more reproducibility? And you see the top answer always is a better and more detailed method sharing would greatly improve reproducibility. So that's what we're here for today, to teach you some of these practices on how to do that. I mean, you see the other things obviously also play a huge role, but methods are so crucial. So this is why I love Methods Week. This is why we are here for, right? Next, please. So before we go into the nitty-gritty, let's just say everyone starts somewhere. Every little bit helps. No one's perfect. You don't have to adopt all of these best practices at once. But if you do at least include some of them in your daily practice, it will not only save you time and be good for you in the long term, it will of course also help other people who are reading your research, right? So next, please. All right, let's go to data management and data sharing next. So data management is the absolute cornerstone of science. When I have students coming to my lab to do their undergraduate or master's training, I always let them go through a data management class a little bit with me. And so one thing that they learn is kind of this kind of thing, the naming of things. So maybe you're familiar with this thought. You have like, you labeled your data, you labeled your files, and then you're like, oh my gosh, what did I call it? Was this thing? Is it the mutant one? The wild type? Was my raw data, right? So sometimes there's a bit of confusion on your data storage. Next, please. And so in order to avoid that, it's good to make a plan, a data management plan. We come back to this in the end of the talk, but there's tools to help you do these data management plans. And universities also now have research staff or personnel staff that helps you develop these data management plans. Next, please. So when we look at the data life cycle, there's this cycle of you create data, you have to process it, you analyze it, preserve it, you give access to the data, and then you maybe find other data sets, you reuse it. So it's this endless circle. However, before you start a research project, you should think about what kind of data will I produce as a part of my project. And you can just take a piece of paper and list this out. And then you have to think about, okay, I have all of these different data types. How will I organize each type of data? How will I document it? How will I standardize it so my colleagues who are collaborating with me are able to use it? How will I store it? How will I protect it? So maybe I work with sensitive data. How will I share it? How will I archive these data? So that's already a long list of things you have to do. But it's good to take the minutes up front before your actual project starts to think about these things and really write it out what you want to do. Next is important to think about, specifically in collaborative projects, who takes responsibilities of carrying out all of these activities listed above. So usually institutions, universities have people who do the archiving for you. You can depend on the storage of your institution. So these kind of things you can outsource, but it needs to be clear who is responsible for these tasks. And the next thing is also needs to be clear is when will these activities take place over the course of the project and beyond when the project's finished. So who takes over the lead? What's going to happen with my data after I collected them, analyzed them, published on them? And then super crucial, there's not much information if you only share your data. You have to have the right metadata also. And metadata are data about data. That's something we're going to talk about later on in the presentation. Next, please. So a data management plan is exactly doing that, making you structure these kind of things. Oh no, something happened to the presentation. Nele, we are out of the...
Sorry, I am trying to get it back in here. That's
All right, so what we want you to do is to develop an informative directory structure. So when you store your data, you're going to have some sort of file structure, right? So it's good for each project, if you're a PhD student or you're maybe a postdoc or someone who has several projects, for each project to have a folder where you store everything together. So keep your research materials together, have a folder then, a subfolder for the methods, for the raw data analysis scripts, for the manuscript, a README file or an electronic lab notebook link. Yeah, so you should have an informative directory structure. We're going to go to questions in the end then. And then so for each of these different folders, you have specific content in that, right? So for analysis, probably, I mean, I'm a postdoc, so if like I maybe use the method in 1 year for analyzing my data, and then next year comes and I'm like, okay, I'm going to use this method. So you see here, there's like a progress also for your analysis methods. Maybe you use different ones, but you should all have it together in one kind of huge project directory. Next, please. The second thing they need to think about and keep in mind is you always should keep your raw data and not touch it again, really. So don't, like if you use Excel sheets, for example, to store your data in, make sure that they are saved in a, like, for example, comma separated value file, and then only make a copy of that when you want to work with them. So do not like open your raw data, whatever, however you're working, and then kind of make the mistake of accidentally changing them. Yeah. So you always need to do a copy of that to really keep them as like after they're cleaned up and correct, to not touch them and not like alter them out of accident. And always back up your data. So we suggest to do it 3 times and synchronize. So maybe the cloud storage of your institution, a server space, and maybe your personal drive or your computer. Next, please. Let's talk about how to name files. So if you can in your head quickly go back to how did you call the last file you generated. We had in our teaching today poster presentations, and one of the posters was called poster.pdf, which is great, right? So you know what it is, but there's no name attached to it, there's no context, nothing, right? So do you have rules to name your files, right? You see here in this cartoon, next on the next, like right next to this writing, you see that file names can get very long, right? And naming something final is probably sometimes not the best idea. I mean, it's good if you have submitted the manuscript, it's peer-reviewed, everything's out, then you can maybe do the final tag, but before that, you should go with versions. Next, please. So we recommend that you have some file naming conventions. So you have some rules, how you name your files. It doesn't really matter which rules you take. It would make a lot of sense to follow some of our suggestions here. So to include the year, month, date format for your files, use meaningful abbreviations, have some sort of group identifiers, and having known like, okay, who was like touching that file last. Be consistent with it and use version numbers. So do versioning. So people who do programming, they know all about versioning usually, but if you don't, you probably don't know about these things. So next, please. There's an example of a very long file name, which is though very specific. So you can see we have the date, we have the project identifier, we know what kind of data is in it, who touched it, which version it is. And at the beginning, it might be a little bit tedious for you because it's so long, but you get used to that. So it's usually just a matter of being used to these kind of things. And I usually share a cartoon here also, but I just want to quickly explain. You should use underscores or hyphens to kind of separate these different names here, or you just capitalize things, just like doing a hashtag, right? So do not use spaces in your file names, because if you have to load them into any kind of computer system, then this will not work. So no spaces, but hyphens and underscores. Next, please. All right, let's go to electronic lab notebooks. Next, please. So the typical notebook, I'm a biologist, we work in the lab, I study plants, so we don't really use high-tech equipment. We use the good old paper notebook for the most part because we don't want our tech stuff to get dirty in the soil. And paper lab notebooks have been around for quite some time, since the 15th century, and you can see here, there's a nice example of Leonardo da Vinci's notebook. But of course, I mean, this one's preserved because it survived all the fires probably that happened in the last 500 years, but short, a lab notebook is good for disseminating ideas and findings, and it's also a legally binding record to protect your intellectual property. But lab notebooks, which are physical and not searchable, they can easily be damaged, misplaced. Students take them home with you, and you need it as the supervisor. These kind of things happen, right? And they're hard to share with collaborators. Next, please. This is why it makes sense to have an electronic lab notebook. So these are searchable, you can export your data, you can share them over links. You often have access globally to them. So if you're going to some sort of other kind of conference or whatever, you can still access that. There's often mobile apps. You can embed high resolution images. Next, please. And there's a lot of different options of these, but most of them have a similar functionality. So this is a bit of an old screenshot of lab archives, but it tells you that already you have on the left some sort of structure, some sort of folder structure. You can copy paste methods in there relatively easily. You have some office compatibility option. You can search it. You can do all kind of things with it. And so organization and kind of sharing your photos also of the things you took. So this is here a PCR gel electrophoresis picture, obviously. So these kind of things, you can just attach that and then move on with it, right? And then each entry is dated, which is good if you work with the industry, for example, because they want to know sometimes also these kind of things. Next, please. So there are a lot of options. There are a lot of paid for options, only paid for, but most of them come with a free version you can try out for a certain time. Then there are also open source electronic lab notebook version software out there. Some institutions like MPI, some of them create their own electronic lab notebooks. And then there's also free things like the Open Science Framework, although in my book, I wouldn't consider that a lab notebook. I think the functionality is not really there, but of course you could use that. Yeah, next please. And so we're not going to make a recommendation what you should use. That's a question that's coming up all the time during workshops. There's a decision matrix that's archived in Zenodo. If you click the link here and you scroll down on the web page, it gives you this decision matrix. And you have the chance to then check, okay, what functionality is important for me? Like, what features do I need? And then it tells you, like, okay, so these different software companies provide this and that. It's a very dynamic market. And as I said, that matrix is archived in Zenodo, so I'm not sure if it's really up to date, but it definitely helps you if you start out trying to find the right solution for you. Next, please. And it's important to keep in mind that still you have to back up your data regularly. Yeah, so if you for some reason lose access to this electronic lab notebook, that would be really bad. Depending on the research you do, you probably should maintain a physical observation notebook in parallel. And then if you use a free electronic lab notebook, specifically here in the European Union or in Germany, you really have to check privacy and data ownership policies and have to make sure that the servers who are storing your information are in a location where you, for example, you work at a university where you're actually allowed to also do that. So maybe if the servers are overseas, maybe that's a breach of trust or some sort of privacy issue. So make sure that you are like kind of doing everything as like legal as possible. Many institutions now have subscriptions to electronic lab notebooks, so go to your own institution and inquire what kind of subscriptions are probably available. I know that our university has one, so I would start with this one and see if I'm kind of, yeah, if I can work with this and then go from there.
Okay, that means that it's my turn to take over. Thank you, Susie. You are perfectly on time, so I think we are doing great here. My name is Nele. Nice to meet all of you. Nice to see you all here. I'm an assistant professor at Baylor College of Medicine, and I joined R4E at a time when I was really struggling to get a well-established method in the field to work in my own hands. And so that is why I think a lot of you are here today. And so what we're going to talk about now is really how to adopt, how to write, and how to share methods so that they can be reproduced. And so really, if you think about it, the ability or being able to find an optimized method that actually works for your tissue, for your species, for your organism of choice can really save you months. And this is really because you would not have to reinvent the wheel. However, very often, even if there is a method available, we struggle to actually get it to work in our hands. And so just before kicking us off today, I wanted to ask a question in the Etherpad. I don't know if it was added. Yeah, so we have a poll there asking you where you go to find protocols. If there's something that you want to try out, a new experiment that you know, nobody in the lab has done yet, or if they have, you know, you can go ask them. But who do you turn to find protocols? see some activity there. So people are asking experts in the field. Nobody is creating them themselves. That will take some time. Asking their colleagues, asking people in the lab. And then we also have the literature. And so, yeah, those are the typical places where you would go to find. And in general, if if this is a method that is new to the lab, people tend to have to turn to the literature or to repositories right now. And so the main challenge there is that when you turn to the literature and you go to a paper, you go to the methods section there, you often end up in this vicious cycle where the method is not really described in great detail due to space restrictions within the article or the journal. And so you are referred to looking at a different paper. That one refers to a different paper that was published before. And so you end up actually either stuck in against a paywall or never actually finding how exactly an experiment was performed. On a different note, when you look at a published protocol or you find your method and you actually try to do it yourself, by the time that you get it to work, the actual steps and the final product tends to look very different from what the original was. And so then it's up to you to try and figure out what you did differently to end up with this completely different results. And so these are just a couple of issues in the field that I think almost all of us have encountered and what we're going to try to overcome today. And so we did ask you in our pre-workshop survey. Thank you all for filling that out. It's really, it's something we see with all of our workshops. These distributions tend to be very similar. Almost all of us have issues when we try to reproduce a method. There are very few of us who have not had issues yet, and those are usually the early career folks who are just starting out. So at some point when you are trying to get a method to work, you will probably get some issues, in having it work perfectly. And that is normal, but we are trying to find ways to actually reduce that struggle. So how do you actually go about adopting someone's method? I thought it would be good to start here today and think through the different steps that you can go through when you are trying to establish a new method. Like many of you said, you go to ask your colleague for help. And so the best way to start out is really to shadow someone who's familiar with the method so that you can observe their find. They will, most people will have a written protocol and they'll give it to you. But then if you actually watch them, you shadow them, you'll notice that there are a lot of small tweaks that really aren't present in their written protocol, even if they wrote it themselves, because they don't think that is important enough to write down, but they often really are. So the best way to adopt a new method is really to shadow someone who knows how to do it. But that's not always possible, right? And so if that is not available, the second best thing is to look for published protocols, whether that is in specific journals that are focused on protocols, or whether that is in protocol repositories. It helps to go look for there are usually different protocols available to do the exact same thing in the same tissue. And it shows you that something can be done in many different ways, but you do really need to understand exactly how it's done. And so here it helps, if this is, for instance, published on a repository, to look at the comments section and actually go look at what people may have added there to explain what made this work for them. The next important thing is to really perform every single step as it is described or as you have observed. And here, I can't emphasize this enough. Details really do matter. Whether you put something at 4 degrees for 2 minutes or you put it at room temperature for 2 minutes is going to make a big difference if you're trying to get an immunostaining to work, for instance. So you have to really look at what is written down or how someone does it and try to take note of as many details as you can. As you go through this, you will probably find that even when you do it on your own, even if you have been as careful as possible, you may still not get the method to work the first time you try this out or the second. And in that at that point, it's very helpful to go back to the individual who either published the protocol or who showed you the method in the first place, and go ask them, look, this is my result. What do you think may have happened? If this is you reaching out to a published author, it may take some time for them to get back if they actually see the paper, if they have the knowledge of that method. So this may be a bit challenging, but if you can go back to someone in your lab or in your institution who has done it, then that can be a bit easier. So in this case, what I found very useful is at the moment when I was trying to optimize my method and I had gone through all of these steps and I really couldn't figure out what was going wrong, why my tissue wasn't sectioning beautifully as I could see in everybody else's papers. And so at that point, I just tried to ask around within my institution. I knew there were other groups who were working on the same tissue. And so I just went over there and I went to ask, hey, I'm trying to do this one experiment, and for some reason, my tissue is tearing and shredding, and I'm just not seeing a nice section on my slide. So how can I fix this? And then I was able to shadow someone and actually see the differences, and then I got it to work. So asking around is really, I think, the biggest trick. Being able to see someone do it, that will save you the most amount of time. And so then at the end of it, you will have found some differences that were not listed in your original protocol that you started out with. And so at that point, it's really important to identify those differences and actually write your own version of a protocol that includes all of those details so that somebody else or yourself, 2 years later, if you want to repeat it, doesn't have to go through that whole process again. So at that point, it's really best if you take the time to actually write down a new version of that protocol that includes all of those details that you figured out are crucial elements. And so this is, one of the ways that you can adopt a new protocol into the lab and that you can actually make sure that other people who come after you will be able to repeat that and use it for their own studies. So there are multiple ways that you can then share that protocol. The best way would be through protocol repositories. There are several free options available. The important thing here is that you look for options that have versioning access. And I saw someone listing Prospero and OSF. Those are also very good options depending on what field you are in. So by now there are multiple options for you to publish that protocol, whether it is as a protocol itself, whether it is now as part of specific journals that are focused on publishing methods. There are different options for you. We tend to like protocols.io for several reasons. There is a comment section there. They have versioning control, and you can actually fork or copy the protocol into your own workspace so that it actually runs for you. And then lastly, you can mark, if you have tried this protocol out, you can actually comment on whether this protocol worked for you or not. And this can really help you identify out of the I don't know, 500 protocols that are out there showing you how to do Western blot analysis for a specific tissue, for a specific organism, which one is the most likely one to also work for you. So that's something to look out for. And so I've told you that it's important to actually write reproducible protocols, but how do you actually do that? How do you go through those motions? What's important to include? I think we've already hammered on the importance of being as detailed as possible. And we're going to drive that message home with a short protocol drawing exercise. So this is going to take us about 5 minutes. I would like, if you have a piece of paper close by to you, grab onto it, take a pencil or a pen or a marker, whatever you have laying close to you. We're going to, like I said, this is going to take about 5 minutes. If you want to step away, if you need a bathroom break, feel free to do so. If you want to join us for the exercise, then I will start it in just a second. So grab that piece of paper and you can get started. I'll start my timer over here. And so the goal is for you to follow these steps, and if you get to the end and you have an idea of what we are drawing here, what this protocol is asking you to do, for now, don't put it in the chat or on the Etherpad just yet, so that you don't spoil it for others. But let's see if you can actually figure out what we are asking you to draw here. We have about a minute left. It looks like two people have more or less finished. We have a guess, although they may be some morphs. Does make it easier to adjust if you've seen what has been guessed.
So I've done this so often, but I'm always putting myself through the initial thing, and I don't know. Don't understand half of what's written and what I'm
I
supposed to do
I I
actually.
still have the same. I'm like, what do you mean 8/10 of an
Yeah,
oval?
what is an eighth of an oval?
Eight
What?
tenth of an inch of what do you
Yeah,
mean?
what's the context, right? So methods, so context dependent.
Yeah.
We tend to
And
assume.
we are oh sorry, go ahead.
No, I'm just saying we assume other people have the same knowledge that we have, right? But specifically in across different countries, that's really very, very different. So
Yep.
context is key.
Things that seem obvious to you right now may not be obvious to other people or even to yourself 2 years down the road. So this is really a very important aspect. If you take anything home from today, it is really that details matter. So if you are somewhere where you feel comfortable turning on your camera to show us what I like those black things in the middle of his mouth or their mouth.
Yeah, it was something with squares, right? So I didn't know what that was doing. It also doesn't have lines, right? So I mean, oh,
Ooh, on a
Alex.
post-it note, nice.
Ah.
That turned out pretty good, Emily.
That's good.
Good job.
Great. Yeah, good job.
Yeah, so just to drive the message home that how something is described really matters and that it is often difficult if you are not familiar with this method or you know with whatever is being done that it can be very challenging to actually correctly interpret how something is described. And so one specific protocol really can lead to many different results. And these, in this instance, look mostly like SpongeBob, although I don't know if I would recognize it out of this one. But I can tell you I've tried this in the past and mine really, I mean, we're nowhere near recognizable. So this is really just to drive that point home. And what we found when we asked all of you before the workshop, if you had tried to reproduce a method, and if you had, or like if you had issues with reproducing a method, what the issue actually was. And you can see here that the vast majority of you found or struggled with the fact that there were not enough details to describe the steps. And that's kind of similar to what you found with the previous drawing exercise too. You have to really go into as much detail as possible. And so we wanted to take some time today to walk you through how to actually write a reproducible method. And the trick here is to try and think of each method or each protocol as a standalone small scientific publication. And so you start with a short abstract that puts the methodology in context. This I've told you many times before, so I won't read it out again. You can include some expected results. What would, if you follow this step by step, what would you expect to see at the end of this method? Obviously, you want to tell people if they are working with some toxic or dangerous chemicals that there are certain things they should watch out for, that this is not something you can pour down the drain. Or stick your finger in to feel if it's the right temperature, and any kind of notes or tips and tricks that you can give them. And then, like we said earlier, it's important to then share that protocol, and ideally through whatever method you choose, look for one that allows versioning. Because I can tell you, if you do your Western blots over and over again, for every antibody, it's going to have slightly different versions, right? So you can actually make different versions for each antibody or over time if you change things. Okay, so to help you out, we created a template. A template protocol. We put that in a regular Word document, but obviously, you know, as you start working on it, you can put this in your own platform of choice. And so this one really walks you through all of the different steps that are important for someone else to try and reproduce it. And so, like we said, that first short abstract version of it You try to describe your method, include detail. This is a protocol for a Western blot. Doesn't help anybody if you explain this is a protocol for a Western blot for tissues obtained from Drosophila, for instance, for the brain of an adult. Those, if you add those kind of details, then the user will actually understand if this is applicable to them or not. Um, After that, the next important thing is to be very detailed in every possible reagent that you've used for that method. And here I just listed a couple of them here. Obviously, there can be others, but try to list all of the different elements that you will need for this method. Explain where you got them, what the product number was. But the problem with the catalog number is that if whatever company you bought something from, either, after a while that no longer exists or gets bought by another company, those product numbers can change. And so there's an alternative to the catalog number or product number that's called RRID. And these are numbers, research resource identifiers that will not change. It's a unique identifier and it is linked to a specific research reagent. And so I would really recommend you to, for your specific reagents, go look whether they have an RRID. And you can do that either through the SciCrunch website. You can click on the link in the chat, or you can check your favorite repository where that reagent came from. If you created a new resource like a plasmid, for instance, or an antibody, and it doesn't have an RRID, or if you have a reagent that is pretty well established in the field, but it doesn't have an RRID, you can actually request one through the SciCrunch website. And I know there was a workshop on RRIDs earlier this week, so if you didn't manage to catch that one, you can still go look for the recording to find out a little bit more information. Here are just a couple of examples of what that looks like and how, for instance, if you work with mice, you go to the JAX website, you can find the RRID there. For plasmids, for instance, with Adgene, you have your RRID over here at the bottom of the page. For antibodies, there's actually something called Antibodypedia, where they are validating or listing antibodies that have been validated by others. And so you can see that that is listed there too. So those are just some ways that you can make sure that someone can find the exact reagents that you used for your protocol. The next step is to really, like we hammered on already many times, describe every single step in as much detail as possible. Here it's helpful to include some photos or pictures, some videos or diagrams where possible to explain or visually show how something was done. Make sure you include what the timing was for any incubation step, what the temperature was it rocking, was it not rocking, was it just sitting on the bench? And an important thing or a very helpful piece of information is to tell someone when you can actually pause an experiment so that they can plan this into their week and see, okay, if this is, a 2-day experiment, where could I pause it so that I can actually get some sleep or food. Then what I also find quite helpful is to include some details about what you would expect to see and what important controls are to include in your experiment. What's a positive or a negative control that you can include to make sure that it's working. And then lastly, it is, I can tell you that it will be very much appreciated by anybody if you can help them troubleshoot. So what are sort of the issues that you have had with this method and how can you overcome them? Does this specific liquid crystallize? And if it does, then, what does that mean and what are you supposed to do? Like I said, safety information, very much appreciated. And then any other possible tips or references that you think may be helpful. And so if you go through those steps and you fill those out, I can guarantee you that that your protocol will be much more reproducible than if you had just listed, like your individual steps very briefly on a Word document by itself. So as we mentioned, the template for this protocol is freely available to you. You are welcome to modify it, and you can find that on the slides and maybe also in the slide deck. So, like we mentioned, once you have actually written this protocol, it's very important to then actually share it and try to save your fellow or future lab mates from having to go through this exact miserable experience that you may have gone through. So save them and save yourself in the future. That's the part we had for methods. I did also want to follow up with a little bit more information about the importance of wet lab reagent sharing. Like I already mentioned earlier, it's very important if you want to reproduce something to actually be able to use the same reagents. And this tends to be really challenging. Because I think many of us can relate with the status of the -80 in the typical lab environment. And so if you go to ask a reagent from another scientist that you have seen in a publication, it actually most often, not most often, but it is quite difficult to actually obtain that reagent, even if the scientist, like has all the goodwill in the world. For them to actually go and dig it out of the freezer may not be that simple. And so there are a lot of problems with the availability of these different types of reagents. For the person requesting the reagent, they have to wait to actually get the reagent from the corresponding author. That slows them down. And sometimes they also have to recreate it themselves. And as we talked about before, that can take quite a bit of time. For the person who is actually trying to send the reagent, the lab itself doesn't necessarily or doesn't always have enough resources to properly track and store all of the different reagents, to actually authenticate them before they send them out, to make sure that if it's a plasmid, that it's actually containing the right insert instead of, like the one right next to it that was completely different. And to legally distribute all of these reagents, to navigate the shipping, pay for the shipping. So there you can see there are really a lot of different issues with every single one of these steps. And so one of the solutions, or one of the best solutions there, is to deposit your reagent at a repository. Like we talked about before, like Addgene, like Flybase, like JAX. This helps you get much better and faster access to materials. They have been authenticated, so you know that what you are getting is exactly what it is or what it's described to be on the website. I would recommend you to validate that yourself in some way before you start, because errors do happen. They are properly curated and standardized. You get all of the information you need, and you will get it faster. Here are just some examples of different types of repositories. You can go back and look at this slide. You can add some in the Etherpad too if there's one that you use more often. But in general, when you are going through thinking about your own reagent that you created in your project, and you want to share it, you want to actually make sure that other people can easily find it and use it, you have to make sure that you very carefully document how the reagent was created, and that you authenticate it before. Here it says regularly, but in general, you have to make sure that you authenticate it before you deposit it at the repository. For yourself, as you recollect the image of the stocked freezer, try to be as organized as possible so that you can find your own stuff, or if you leave the lab, that you can actually tell people where to find things. And when you name reagents, just like Susie was telling you earlier for files, make sure you have consistent rules that you follow within the lab so that other people can understand. It's not just plasmid 1.4, but it has a little bit more description for people to understand what it is. And then, include the RRID if you have it. To help you organize your own -20 or -80 storage space, we also created a template that you can use to just fill out the names of all of your different boxes that you have, whether it's -20 or -80. You can modify this based on what your freezer space looks like. This is showing you the organization for a rack like this with different shelves, like different I don't know how you call that, different layers within there, different drawers, I guess. You can modify. You can see here it has three shells. In this example, it has four. You can put the name or the label of each individual box on there. And then on the next tab, you can also actually put the different, if you, for instance, have a 9 by 9 freezer storage box, what are the different samples that are included in here? This, I can guarantee you, it takes some time to set up from the beginning, but it is incredibly helpful down the road. So just to summarize, I hope we were able to convince you that it's important to actually share your reagents once you've developed them, that it helps others, but also yourself down the road, as then they can get cited. Make sure that you document the key details about the reagents that you are using and deposit those that you make. And that just makes science all the more reproducible. And I think that's it for my section. So I'll pass it on to Alexis now.
Hi, nice to meet everyone. And it's been great so far. It's been a great session. I'm Alexis. I'm a postdoc at the University of Utah. And I'm going to talk a little bit today about data and code sharing. And just to like have a odd note, there was a little earthquake at the beginning of the session today here. So I had some in and out times earlier. So hopefully no more earthquakes, but it's a little bit exciting.
Yeah, hopefully that's it for today.
Earthquake for the day. But yeah, so if I'm like looking out the window, it's because I'm a little jumpy. You can go to the next slide. So we've had a really good conversation of how to think about sharing all different aspects of your research to make it a bit more reproducible. Uh, uh, and another part of this is to think about sharing the data and code associated with it. So we've talked about protocol sharing, we've talked about reagent sharing. Those parts are both important parts about generating the data that is used in whatever your scientific output is. But the data is something else that is critical, especially at the publication stage, to better understand and to be able to reproduce the work that's been done. So, what to share? Sharing the data and the code necessary to validate and reproduce the results of the work that's being shared. Other code that might not immediately spring to mind is code that would be valuable to individuals who would come across the data later and be interested in reusing this. I think the second bullet point is particularly helpful if there's data that doesn't quite come to a natural publishable conclusion, but might still need to be shared as you've kind of come to the end of a project, but hasn't necessarily had a narrative come together. Um, and then data and code that can't be easily regenerated. So sometimes you might be able to share the code that generates simulated data without needing to share the ultimately simulated data itself. But if you can't regenerate that data just with the click of a button, then that's data that should be shared. Why share? I think more and more as we're kind of growing to appreciate the need for more transparent science. It's a requirement of your funder or your publisher, which has pushed a lot of scientists to better understand all kind of parts of the research, transparency, and reproducibility. But it's often required by a funder. But it also has benefits to you as the researcher. There's citation benefits for the work, and it preserves long-term access to that data. So as you go on your career, you might find yourself like, what was even in this project that I did 8, 10, 15, 30 years ago? And so being able to even help yourself if you want to, in 20 years, do some sort of synthesis work, be able to find the data yourself or have other scientists that might come after you that are interested in synthesizing the work in a single area or across areas, being able to find and combine the data in that way. How you share may vary by the particular type of data you're working with, but generally the best advice is to choose open, persistent, and non-proprietary file formats. And that's kind of a large way to say something like, if you need to pay for a software license to open a file, it's better to use a file type that doesn't require that paid service. So for example, you might be able to store the same type of information in an Excel sheet and in a CSV, a comma separated values table. And storing it in an Excel sheet means that whoever you're sharing it with needs to have an Excel license to open the file. But if you share it as a CSV, you could open it regardless of what computer system you're on. This is true for many types of files, and you'll know a bit better in your own field, kind of what the open formats and closed formats might be. Another important part is to create and share documentation. I saw that a lot of you indicated that metadata was something that you were excited about and wanted to learn more about in your pre-workshop survey. So we'll talk a little bit more about this, but sharing that metadata and that documentation is an important part of making sure this data is reusable. If you're using data that's been shared previously, including data citations and noting kind of the provenance or where you got that data from is helpful for anyone who's looking at this data downstream. And we'll talk through creating metadata. So next slide. So data sharing so this is like a cool plot. I think you'll see that on the x-axis. This is the year that the first URL that this URL was first published, and on the y-axis you've got the percent down. What this means is like as time goes on, the URLs that are available that were originally available start decaying. So you might be across your daily life coming across links that point to page not found links, or that point to empty dead spaces on the internet, point to sites that have changed their names. So over time, the links that are inside of abstracts or inside of papers, or just even generally on the web, are less and less able to point to what they originally meant to point to. So that can make it really challenging to follow the line of links back to find what was originally cited in a paper, or to find any information relevant to kind of what was put in the paper. We see this also in kind of supplemental figures or supplemental data, where the supplemental data that was published with the paper used to be linked or used to be included in the document, but over time that linkage has been lost. So it's very challenging to kind of pull together all the pieces of code and data and even just general URLs online in order to kind of keep them with the original paper. So sharing the data in some persistent repository can really help. Perfect. So how do we get through this? A data repository can help. Data repositories provide things like persistent identifiers, a DOI, which you might have seen before, is a unique and citable link that doesn't change even if the underlying location that it points to does change. So this can let you have a link that stays persistent, can have different versions, but is still usable by whatever used it in the paper or used it somewhere else. Data is usually also published with a license. Data and code both are. And these help users later understand what types of guidelines there are for using or reusing the data in the future. There's a lot more information about this that we won't be able to cover here. So these links you'll be able to find in the slides later to kind of go through what the different options are for licenses to help understand what your downstream users might be able to use for. Next slide. And when you're looking for a repository that you put your data in, there's many different options. You'll start to become familiar with them as you're working in a new field, or you might have come across many of these already. There are many general purpose repositories that are listed here: Data Dryad, Figshare, Zenodo, and Open Science Framework. Data Dryad usually costs money, but is free to access. All of these are free to access. But Figshare, Zenodo, and Open Science Framework have systems where you're able to put your data on them for free. They're generally multi-purpose, so you can put all different types of data on there. There are some restrictions if you're working with like very special or specialty formats or extremely large data sets. So in addition to these types of general purpose repositories, there's also specific repositories that might be specific to the type of data you're working with. And sometimes there are additional access requirements that need to be taken into account. If you're working with data that might be protected in some way, like related to human health or related to, something that might not be able to be shared as publicly, but there are usually repositories as well for these types of data that have controlled access to this type of data. So next slide. When you're thinking about how best to share your data, kind of the guiding principle is making your data as fair as possible. And FAIR is an acronym that stands for findable. Is your data somewhere that you're able to show someone this is where it is? find it on kind of the map of the internet? This is where you could find it. Could anyone else be able to discover it with information about the work and what's in it? Is it accessible? Are people able to meaningfully interact with the data and use it themselves? Is it interoperable? Can you use it on different types of computer systems? Can you use it across different types of system architecture? And is it reusable? Does it have the necessary information in order to be used in some kind of meaningful way? So each of these steps has lots of different layers to it. And many people, especially in the information science world, spend a lot of time thinking about each of these steps in a lot of detail. And I think a thing to start with is to know that these are kind of the goals to shoot for and to start thinking about how you can improve, even in small ways, your data in each of these different steps. Next slide. One of the biggest keys to having fair data is to have metadata. Metadata helps kind of across the board. It can help your data be more discoverable because of how a lot of the repositories work. It can help with your data, especially being more reusable by providing the key pieces of information a user might need. And it can help with that accessibility by providing key information about what is in the data that you're looking at. So metadata is key. You might think of it as kind of the guide to the data you're looking at. What is it? Who generated it? Why was it generated? Where and when was it collected? And how should you use it? Or how can you reproduce it? I think that the XKCD comment here on the right kind of hits a lot of the topics we've been covered here, that there's all of these different file names. Who knows what they mean? But even if you have really intelligible file names, the contents of them might be something that you named the columns 30 months ago, and who knows what's in it now. So metadata can help you and can help your downstream users and viewers understand what is actually in the data that they're looking at. Next slide. Okay, so one of the key parts of a metadata package is kind of a README, which you might know about from looking at a GitHub, but a README is just kind of a fancy way to say a landing summary page. I think of it as kind of the abstract for the code or the data that you're looking at. And this provides information about the title, investigators, who was involved, where was it, the institution, and when was it, what were the dates of the project. It usually gives a short description of the data collection and the generation process for the code or for the data. This I think of as like the abstract, a brief description of the data processing and analysis, another part of this data abstract, and a description of what each folder or data file contains. My kind of shortcut to getting this description of each file is I use the tree command in Linux that spits out what the tree of all the files is in the folder that I'm looking at. And that gives me a sense of what are each file that I need to explain. Because that'll tell me like what is even in this folder that I'm trying to describe and helps me keep track of what's there. It can even give you, when you do this kind of tree function on a folder, the date that each file was created as well. And this is something that you want to keep up to date over time. If you are someone who has a great file naming scheme, or if you're picking up on the one that was taught earlier in the session, it can also be very helpful to give file naming scheme overview for what do people expect to see in this package of code and data. Next slide. Beyond the README, there's usually attached to each data file a data dictionary. And so a data dictionary is an orienting document that, as we were talking about earlier, helps kind of understand the types of things you expect to find in the data file. What is in each column? So you'll see here kind of mapping from this data file to the data dictionary that each column is represented in the file, and it tells you what is the name of the column in the language that's used in the data file, employee underscore ID. What type of data do you expect to find in here? In this case, it's an integer. I like to provide this data type in both machine language, like integer, character, floating point, and also in like plain language that you expect to find in here, a number or text or something like that. I think it's helpful to have kind of both in a lot of cases. And then a description, like what is the meaning of this number? Numbers can mean lots of things. Is this an ID number? Is this someone's height? Is this, each thing? It can also be very helpful here to include If it's a code set, like you might have something like sex, you'll see or sex or gender, like they have down in this column, that gender is a character that you expect only one character for, but it comes in only a couple flavors. The only options here are M, which is for male, in this case, F, which is for female, and null, which is unknown. So this is a good example of providing all of the expected types of values you might find here. Some people might code male and female as 1 and 0. Some might code it as the full word in capitals, in full word in lowercase. And null is helpful too because it tells you whether a missing value means that the participant maybe didn't want to report it, whether the participant is non-binary, or like what does the missing value actually mean in this case is important piece of information for your users to know. So what does this look like in a directory? So as we were talking about, this is usually something that's paired with your data directly. So you'll have the README as kind of an overview, each data file and or code file that it's connected to, and then the appropriate data dictionary that goes with it. In this case, the data dictionaries have the same name as the data file, but added with the underscore dictionary. It's very helpful to match them in this way so that you're able to see for your data file, the associated dictionary will be that name plus the dictionary. There's a lot more resources out there, so this link can take you to some of them because your metadata strategy will evolve as your data strategy evolves as well. Next slide. One, I think, of the key most important thing, if you only take one thing away from this session, is making a plan. A plan will help you, it will help everyone, but most of all, it will help you, I think, next week, 2 months, 2 years. Making a plan now will help you be able to understand what was going on, what was happening in your mind. There's that common coding joke, like this code I used to know, only God and I used to know what it was, and now it's only God. If you're documenting your work and making sure that it's reproducible and transparent, hopefully it will remain both you and God and anyone else who comes across your package. So starting now and as soon as possible can help you build a robust plan. And I think especially as you're kind of moving through your career, it's not too late to start the data plan. Now is always better than next week. So being able to start at any point is always going to help. And you can always make incremental changes to your data plan, your protocol plan to help you be more transparent and reproducible across your whole kind of research life cycle. Okay, next slide. Which I think is a good transition to the idea that reproducible research practice helps you organize new experiments, understand results, share these results, share techniques, share reagents, and accelerate your science. And I want to really reiterate that you don't have to be perfect at all of this at once for it to be a good opportunity to make a small change in a positive direction in your research process. Any change will help future you and will help future researchers that come across your work be able to work with it more effectively and to accelerate your own science as well as theirs. So the tools that we discussed here give you a framework and kind of give you the search terms to know like this is what you're pointed towards and this is how you can think about saving yourself time and resources moving forward throughout your career, but also throughout the duration of any project you're working on. So being able to take these tools and kind of move with them forward, I think is one thing to take away from this session. Next slide. So next steps. If you want to answer in the Etherpad, what is one thing that you can do today to start making your research more reproducible? And we can talk through some of these as we go. I know one thing I am always trying to do. I recently started a postdoc, so I'm trying to be really cognizant of my file structure, which we didn't really touch on very much today, but making sure that my file structure is interpretable really is something not just file names, but where are they is helpful for me.
I think those are good opportunities, right? When you start a new project or a new experiment or a new position, those are good. It can feel overwhelming to have all of these tips and tricks and try to apply them all at the same time. But it's very helpful to just say, okay, I'm starting this new project or this new experiment, or I start my postdoc or my PhD or whatever it is. Let me now try to implement some of these differences. And Alexis, I'll ask you in the meantime, someone asked if you can please share again what the tree function is that you talked about to show, what all the different files are that are in the
Yeah, I
folders.
just added it, added
Great.
the Wikipedia page for it. There's different versions for each type of infrastructure on Windows or Mac, whatever. So it's helpful, but the Wikipedia page has like even a picture of what the output looks like, which is nice.
Yeah, that's a great idea,
Yeah.
actually.
Yeah. I just
And
went
it's
for the
also,
print screen.
yeah, because if you're also when you're used to the Windows kind of like how Windows is working, it does not come with functionality like the Linux systems, for example, do. So with Linux, I personally find we have like a more kind of intuitive structure, and it's kind of so I like the order there very much. But this, yeah, this function is super helpful.
Yeah,
So,
I honestly even just put it on my metadata, read me the tree and what it looks like. So it helps me even
Yeah,
know.
oh, that's
I would
great.
agree.
Great idea. So if you can please do us a favor and fill out the survey. So Nele had hinted on that.
Yeah, I
Now
put
we're
it in the
going to go
chat
over
and
to,
it's also on the Etherpad.
yes,
Great.
exactly. We're going to also address some questions. Um, so there was one that didn't make it into the pad because some people have issues accessing the pad. So let me just go to a question that came up in the chat. Would AI tools play any good role in protocol design, exercising, and sharing? Any thoughts and some helpful ones? So there was on the Open Science Conference last year, which was a global conference on open science and AI. There we discussed this kind of topic, and definitely AI tools are helpful for a lot of the metadata stuff. So the outcome of that session. So if you're interested in that, you can search for Open Science Conference, which was in Germany in Hamburg by the ZBW. So that's a research institution of economics, and they organize this Open Science Conference each year. And the outcome there was there's not like the one AI tool currently that's helping us with this. And unfortunately, people who tried it out still had to check or still had to do manual labor to check if the metadata are really correct. So there are some, it's a kind of the short answer to that is 100%. They are going to be helpful moving forward. Currently, there's not, as far as I know, the one tool that's helping us with this metadata. And so until that is available, please do that on your own. Also to know as a human kind of researcher, like what you really need, yeah, for the metadata. And there are metadata schemes. We didn't really touch on them. For each disciplines, there are these metadata schemes, so you can search for a discipline, what the metadata scheme is maybe for your specific field you're working in. So please go towards them first, and then I think you should like write it out yourself to see like what kind of level of detail is really necessary for other people to be able to work with your stuff.
Yeah, and I mean, you can already use some hacks in the meantime. I think Susie is absolutely right. It's not available just yet, but what you can look for is to use things like ChatGTP or Copilot or whichever version is your favorite and is allowable at your institution. To look for for instance, what antibodies have been shown to work in my specific tissue, in my specific organism to identify, I don't know, a certain subtype of neuron, for instance. That actually already works fairly well to find publications that show a specific, I don't know, paneuronal antibody, for instance, that might work well for what you're trying to do. We have another question to ask if we can use Wikidata to store and share the data. I'll let Alexis answer that one.
That's a good question. Wikidata is great. I love Wikidata. It is a great place to find certain types of structured data, but it's not usually the right place to store multi-purpose research data. There is actually quite a lot of restrictions on Wikidata on what's permitted to be stored there. So it might be the case that what you're working on is a perfect fit. Like I can imagine taxonomists often use Wikidata, but often it doesn't allow for the supporting data that might be used that has been important to like finding one specific data point. Um, so it depends. It really depends on whether your data is the right fit. And I think this is a more general point that not all repositories are good fits for the data that you're using. That's what's nice about some of those general purpose repositories that I went through early is that they're open to lots of types of things, but Wikidata might be a good fit for your work. It might not be. NCBI's sequence read archive might be a perfect fit for your data if you're working with sequencing data, or it might be a terrible fit if you're working with geoscience data. Decentralized data hubs, some of them are amazing, but I think this is actually like kind of in line that some decentralized data hubs are going to be a perfect fit and some are not going to be a perfect fit. It really depends on the particular subject matter. I'm excited about decentralized data hubs. I think they're cool. I think they're really cool. But it is a matter of making sure that what you're looking for is well matched. And I do have some concerns about the longevity of some of these new types of repositories that they don't always have long-term plans for what their storage will look like moving forward. So, making sure we're not contributing to that downward line that we saw earlier, that things disappear off the web is important, and repositories that you choose for your data should be thinking about this and should actually have places on their site that you can see what they're thinking about longevity for. So being able to see that's important. I think that's sometimes kind of hard to see for the decentralized data hubs, but I do think they're really cool and have a lot of promise.
Yeah,
Yeah, and I
I'm
think you're, oh, go ahead, sorry.
No, I just want to say so, from my point of view, it shows also your involvement in your scientific community that you know where people kind of share their data with. So each discipline also has specific data repositories they lean towards publishing their data sets. So for example, the ecology uh, like bubble kind of in biology, they have their certain like deposit repositories for specific data, and it's very important. Alexis shared that findability is really important. So we have this link for re3data.org, which is a meta crawler for data repositories, where you can find different data repositories that exist for specific data, like microscopy data, or like, I'm thinking of all this biology kind of things. If you are depositing your data on a, let's say, a very niche kind of server, it might be people are not able to find that because for some reason they are not familiar with this. So I would suggest very much to really kind of look towards, okay, so what do other people in my field do? Those who do the good practice, those who do the data sharing, where do they deposit their data? And that's what I would go with first. And this is also what conferences are very, very important for, to really connect with the people in your community and find out where people are doing that, right? So this is how I would start. I mean, depending on where you are in your career, but this is where I would go to. So the findability aspect is really there because you want people to find your data and then also cite you, right? So I would, I'm not saying you should never use like a decentralized hub or anything, but I would be a bit careful with some more like the niche kind of data repositories probably.
Into the newsletter.
Yeah, I will try to find that. We may have to follow up with you after. Oh, you have it? Great. So yeah, so before moving on to some of the other questions, I do want to make sure that as maybe people have to head out a little bit early, that we do tell you that we would love if you thought this workshop was helpful, that we would really love for you to be as involved as you are able to and as you want to. So you can do that in many different ways. You can sign up for our newsletter. You can become a volunteer and help us host workshops like this one. You can request a workshop to be given at your institution or whatever conference. You can actually help host workshops like I mentioned, and of course to help us keep our files and folders on our website. We do need a little bit of funds to keep things running. So we all of the information that you saw today is available to you. They are shared. There are many links for you available, so feel free to use them and modify them as you see fit. If you want to use our slides and give maybe a journal club at your own lab to highlight some of the things that you learned today, feel free to do so. This is just, our standard license slide. You are welcome to modify things. All of these resources are available under CC by 4 license. We definitely welcome you reaching out to us with any questions you have afterwards. And these are ways that you can get in touch with us. And so I did already share the workshop in the chat. And so we do actually have a couple more minutes for questions in case there are any other ones.
Nele, I'm not sure if this is the right link. This gives me an error currently.
Oh, really?
Yeah, I'm just seeing if we did a mistake.
Sorry.
I'm going to check it, and then if it's not working, I'm going to.
Yeah, I just copied it from the main. But
So
the one that's in the Etherpad does seem to. Yeah.
Yeah, sorry for that if it wasn't working for you before.
Yeah. There are different links that Susie and Alexis.
Hard.
Yeah, if you shorten it, I think that's where you end up, right?
Okay, we'll find out. Thank you for whoever, if you took the time to fill them out, we will find your answer somewhere. So thank you.
I can't access, at least I'm not sure if it's on my side, but I cannot.
I took mine down just so that we drove through the same place.
Okay, so we can also stop recording now and chat a little more freely then.
Yep.
For everyone who's still around. So thanks so much for everyone who's here.